My name is Adam Tifford, and welcome to my AP Biology Honors lesson. Today we are going to talk about the movement of substances in and around the cell. To begin, we must know the terms for referring to the inside and the outside of the cell, as well as conditions that exist in these locations. As you can see, the inside of a cell is known as the intracellular space, while the outside is known as extracellular. This is similar to how we refer to space as extraterrestrial or outside of Earth. Next we have the description of the amount of water or salt that is outside of the cell. There are three terms that refer to the different concentration of salt that is outside of the cell. These are hypertonic, isotonic, and hypotonic. Hypertonic refers to a large salt concentration, which causes the cells to shrivel up. Hypotonic refers to very low amounts, which cause the cell to engorge with water. And isotonic refers to there being about the same, or a balance. Next we must understand how substances flow from one place to another. There are two main means of this. Broadly, they are known as active and passive transport. In passive transport, a substance in higher concentration naturally flows to the area of lower concentration. This requires absolutely no energy. Now, I know you may be thinking, what? Now, Professor, how in the name of science does something move with no energy? Well, there is a very simple answer for that. There is a natural movement in atoms that have any sort of temperature that is not absolute zero. This movement in an atomic scale causes motion in the substance. In fluids, this will cause a flow. In this flow, the higher concentrate will gradually place more of the substance into the lower concentrate areas. This means that without energy being added, these two will eventually flow together and become neutral. That is to say, they will have the same concentration of the substance. What you have here is passive transport. To help you conceptualize this, I have prepared an analogy. When you pack too many items into a box, they get cramped and push against the box. When they are released, the box will burst open and spread its contents out. This is much like passive transport. Now to take that analogy and look at it backwards. If you were to play out that analogy backwards, and you were to stuff a bunch of items into a box and hold it there, you would essentially be looking at active transport. This is a type of transport that does require energy. It is essentially the antonym to passive transport. It requires energy once more. It moves from low concentration to high concentration, unlike passive transport, and it does not happen naturally without some sort of intervention. Active transport is when something is basically forced into a location. One example is known as bulk flow. This is when the force moves something in one direction and it flows in that direction in response. A real-world example of this is a river in which water is being flowed. You can see here on the right that this river would be an example of bulk flow. Now that we understand the two main types of transport, we can move into how they apply to movement through cells. There are a large amount of different types of passive transport that are used by the cells. These are simple diffusion, osmosis, dialysis, plasmolysis, facilitated diffusion, and, of course, countercurrent change. Simple diffusion is essentially passive transport at its most basic level. This is when the molecules simply move from high to low concentration and not much more. What is special about this in the cell is that there are only a few substances that are in the human body that can permeate cells just because of how small they are. These are oxygen, water, alcohol, nitrogen, and carbon dioxide. They are all able to do this because the cell membrane is semi-permeable. This means that some things can get in and others can't. This is synonymous to a child at a private neighborhood. There would be a gatekeeper, of course, and they would have to open the gate for most adults to get in, but there are some children that are simply small enough to fit through the hole in the gate, and these would be these molecules. Next we have osmosis, as represented by this picture of the popular cartoon Osmosis Jones. Though this is a part of simple diffusion, it is very important on its own. Osmosis refers to the movement of water from extracellular to intracellular or vice versa. One form of osmosis is known as plasmolysis, and it is what happens when there is a very hypotonic solution around the cell. In this case, the cell will absorb all the water and it explodes like a balloon and ruptures and releases all the engorged water that it had from absorbing all this water, and it essentially dies. This is a very violent death. Following this we have dialysis. This refers to the diffusion of solutes into the cell membrane. This is a process in which semi-permeability of the cell membrane is shown and highlighted. Usually in the case of this type of movement, the cell membrane acts as a filter because only certain parts of the mixture will be able to get through. After this, there is another large concept known as facilitated diffusion, which is a form of passive diffusion because it requires no energy, but it also does not happen naturally. You can see in this analogy pictured on the screen It's really more of a protein that guides whatever substance is going in, the protein being represented by the larger hand and the smaller hand grabbing onto it being whatever substance is being brought into a cell. This happens when solutes are not able to diffuse into the cell simply, so instead they are guided in by the special proteins again. These proteins facilitate the movement. Though it would happen on its own through simple diffusion, eventually the solutes can move through much faster through this method while still requiring absolutely no energy. Finally, there is countercurrent change. This is when two bulk flows occur in opposite directions and the substances in them mix into each other. As far as active transport goes, the cell does not do very much with it, relative to passive transport, that is. It does have a very large purpose, and this is to bring in, forcefully, amino acids and monosaccharides, which are too large to engage in passive transport. These can be used for energy later, after being put into the mitochondria and turned into ATP, as well as fortification. The final way in which the cell takes in substances or releases them is known as vesicular transport. This is when the body uses vesicles, which are essentially fluid-filled sacs. These are used to carry things into the cell very easily. This is similar to using a car to get somewhere. Though the important thing is inside of the car, it is being used as a means of transport. The analogy, of course, being shown on the left and the literal vesicle on the right. There are two main types of vesicular transport. One of these types is exocytosis. In the process of exocytosis, a substance that the cell wishes to move into extracellular space is put into a vesicle, and that vesicle is sent towards the cell membrane. At this point, the vesicle fuses with the cell membrane, and the content that the cell did not want is released. The way that I see this is like a fast food restaurant window in a dangerous neighborhood. They need to get the food to you, but do not want to open the window up completely as to let in anything dangerous. They then put the food on the tray and open the box window so they can put the food out and let nothing else in. It is also similar to exporting, as shown on the screen here. On the flip side, the other method of vesicular transport is a bit more complicated, as it is in itself 3 parts. The other method is known as endocytosis, which, not surprisingly, is when something is taken in or imported. The basic way that it does this is that it engulfs the substance in the cell membrane and wraps it in a vesicle. The vesicle is then put into the intracellular space. To get into finer detail, one way in which this occurs is known as phagocytosis. This sounds like some sort of horrid disease, but in reality is a very important cellular process. It is referred to in layman's terms as cellular eating. This is because this method is very similar to how someone eats food. Essentially, this occurs when the cell engulfs a non-fluid material. It then wraps this substance or foreign life in a phagocytic vesicle. From there, it essentially digests it. Some cells, like the macrophage, use this as a means to defend the body from intruders by engulfing foreign bacteria and killing it in this way. Following this, there is penocytosis. This is also known as cellular drinking. This is when a fluid is brought into the cell by means of vesicular transport. Essentially, the liquid is engulfed, then released into the vesicle, then into intracellular space. It is referred to as cellular drinking because it is a fluid rather than a solid, and therefore would be like drinking rather than eating. Finally, there is receptor-mediated endocytosis. This is technically a form of penocytosis in which specific molecules are filtered out of the liquid by binding to special receptacles. This is used to transport things such as cholesterol about, because once it is in the pit, it is in its purer form, and then can be put into a vesicle of its own, making for much more efficient storage. It is similar to how a coffee filter will filter out the grains, so you can get the flavor of the coffee, but don't have to drink the grains. This concludes my video, as we have now covered all the types of movements in and around the cell, as well as through it. I hope you learned something from this synopsis of the different types of movement through the cells. Thank you for watching this video, and have a good day.